All right, Ninja Nerds, in this video we're going to talk about the Loop of Henle. Now, if you guys haven't already seen it, I want you guys to go into the actual, specifically the kidney playlist, and take a look and watch the glomerular filtration and the proximal convoluted tubule video before you guys come and watch the Loop of Henle video, so that you guys can tie it all together. Okay, so Loop of Henle. There's two parts of the Loop of Henle. So the Loop of Henle consists of the descending limb, which is this part here, and then coming up the ascending limb. So again, let's actually define that again. So the Loop of Henle is broken into two parts. One is the descending limb. And we're going to talk about this one first. And then the second one, the other component of the Loop of Henle, is the ascending limb. Now, before I do this, I want to introduce another term that you guys might not have been familiar with, or you might be familiar with. This term is called nephron. So I want to talk about this term called nephron. So nephron. What is a nephron? Okay, so nephron, you remember we talked about this within the actual glomerular filtration video. It's consisting of a renal corpuscle. So what's a renal corpuscle consisting of? This right here, the glomerulus and the associated Bowman's capsule, right? So far, what's the nephron made up of? Let's put it in like a little, maybe like a little mathematical equation here. We'll say glomerulus plus Bowman's capsule plus, we already talked about the proximal convoluted tubule. So this is another component, the proximal convoluted tubule. So we'll put PCT. It's also going to be the Loop of Henle, so the descending and the ascending limb. So we're also going to add Loop of Henle to this. And then the last part is the distal convoluted tubule. And that's this part that we'll talk about in the next video. This is what's making up our nephron. Now our nephron, again, one more time, is made up of the glomerulus, which is the capillaries, the Bowman's capsule, the proximal convoluted tubule, the Loop of Henle, and the distal convoluted tubule. You know, within one kidney, you have about 1.2 million nephrons, but you know, usually you have two kidneys. So if you have two kidneys, how many total nephrons does that make then? 2.4 million of these nephrons within your two kidneys. That's unbelievable. Okay, so now let's get back to the Loop of Henle. So the Loop of Henle is a component of the nephron, right? So let's talk about the descending limb first. So if you guys remember, what did we call? Let's catch up. We had filtration, the glomerular filtration process, right? After that, we had this process of tubular reabsorption of certain types of chemicals, right? And we had the process in which we discussed tubular secretion of certain chemicals, right? So again, this is called tubular secretion, and this right here is called tubular reabsorption. Now I got to get some numbers in here again. Sorry guys, I really wish I didn't have to do all these numbers, but if you remember, what was the osmolarity within inside of the glomerulus? It was approximately 300 milliosmoles. What was it when it went into the Bowman's capsule? It was approximately 300 milliosmoles. Here's the next question. What is it when it leaves the proximal convoluted tubule? Some of you might be like blown away by this. Some of you might understand it. It's 300 milliosmoles. Now you're probably like, well, holy crap, Zach, you reabsorbed a lot of different salts and bicarb and nutrients and all that kind of stuff. Why would it be 300 milliosmoles? Shouldn't it be less? No. Why? I'm going to tell you why. Out of the substances that you reabsorbed, remember that we had 65% of that was actually sodium? And how much of it was water? Exactly the same amount. 65% of it was also water. If you reabsorb the same amount of water and sodium ions into the blood, don't you have the same amount being reabsorbed? So if that same amount is being reabsorbed, then what's coming into this actual Loop of Henle, this descending, should now be the same as the actual plasma osmolality. So it's 300 milliosmoles. So it's what we refer to it as isotonic with the blood plasma. So this is isotonic with the blood plasma, and then the actual filtrate in the actual Bowman's capsule is also isotonic with the blood plasma, meaning it's equal. But watch what happens here. As we're actually coming down this actual, specifically about renal medulla, because whenever you get to the nephron loop, this Loop of Henle, it's dipping down into the actual renal medulla. So as it's dipping down into the renal medulla, look what happens to the actual concentration of the actual osmolality as we move down. So right here it's about 300 milliosmoles. Then as you move down, it gets to about 500 milliosmoles. If I move down even more, it gets to about 700 milliosmoles. If I move down here, it's about 900 milliosmoles. And then all the way down here, it's about 1200 milliosmoles. If you're looking, what's happening to the actual plasma osmolarity, or just, I'm sorry, this actual medullary interstitial osmolarity? What's happening? It's getting saltier or more hypertonic as you go down the renal medulla. So as I'm moving down this renal medulla, the actual concentration of this actual salty ions are actually increasing. It's becoming more salty as I move down. All right, so now let's talk about osmolality. Osmolality. Okay, if you remember, we said that if there's a high osmolality, that refers to specifically high amounts of sodium and chloride in the blood, right? Or just a lot of solutes in the blood. So we can refer to this as solutes. And then we said that it was a very low amount of water. This is referred to as, when we have high amounts of salt and low water, this is referred to as hypertonic. Now let's do this another way. Let's say we do it like this now. What if the sodium chloride, or the sodium and the chloride and the different types of solutes in the blood, let's say that this is less. And then the amount of water is more. When this is the case, this is referred to as hypotonic. And what would this person's osmolality be? This person's osmolality would be very, very low. But now let's say the last scenario. So let's take the last scenario. And let's say that the sodium chloride, or just our solutes in general, is equal to the amount of water. What's that referred to? This is referred to as isotonic. Okay? So now, we have to take this into consideration. Now that we understand what these terms of osmolality and hypertonic, hypotonic, isotonic means, let's look at this. As we're moving from the actual cortex all the way down deep to the renal medulla, down to the renal pyramids, all the way down to that papilla, what's happening to the osmolality? It's increasing. So what does that mean? If osmolality is increasing, that means there's a lot of sodium and a lot of chloride ions in that area and very little water. Okay. How in the heck am I getting this salty? I'm glad you asked. You see this googly-eyed frickin' guy right there? You see that googly-eyed guy? That googly-eyed guy is a very special transporter. This googly-eyed guy is called a sodium, potassium, 2-chloride cotransporter. Holy crap, that was a lot, right? That was a mouthful. But again, that's what this guy is. This googly-eyed guys are going to be sodium, potassium, 2-chloride cotransporters. Look what they transport. They take sodium, they take potassium, and they take chloride ions. And they transport it all from the actual lumen of the actual filtrate into this actual tubule cell of the ascending limb. And then there's special transporters or special types of channels here on the basolateral membrane. And each one is specific to these guys. So there's B right here. Let's say that's a sodium channel. Let's say that this one right here is going to be a potassium channel. And let's say that this one right here is actually going to be a chloride channel. What happens? Chloride's going to get pushed out. What's going to happen to the sodium? He's going to get pushed out. Now the potassium, some of it, a small amount of it, will actually leak out. But some of it will actually stay in this area, and we'll discuss that in just a second here. But now, what's getting really, really concentrated out in this area then? We're having a lot of sodium concentration is increasing, so a lot of sodium is going to get pushed out here. So let's show a lot of sodium ions increasing out in this area, and that's why it's getting saltier. What else is increasing in this area? There's also going to be a lot of chloride ions in this area. So there's going to be a lot of chloride ions in this area because they're also getting pumped out here too. So what is this? What did I just say? As we're going down, it's getting saltier. Why is it getting saltier? Because these sodium, potassium, 2-chloride cotransporters are actually pumping this, not actively, they're pumping this sodium, this chloride, and some of that potassium out here. So what is it going to be doing here? Same thing. So what will this be pushing out? Sodium. It'll be pumping out potassium. Some of that potassium will actually stay out here though. We'll talk about why in a second. And then chloride is also going to be pushed out. So what's happening to the concentration here? Chloride concentration is increasing. The sodium concentration is increasing. And the potassium concentration is increasing just a tiny bit. Nothing significant. We'll put like a tiny little arrow there. Okay. But either way, nonetheless, what's happening to the concentration out here? It's getting salty and saltier and saltier as you're going down, right? Now, this as you're going this way, it's pumping out sodium, potassium, and chloride. But as this is going down, what's going to be one of the components in here? You're going to have a lot of water in this area. So water loves to follow sodium chloride. I told you that, right? Well, where's that sodium and where's that chloride at? It's out here in that medullary interstitial space. So out here in this medullary interstitial space, what's happening? We're having a lot of sodium ions out here, and we're having a lot of chloride ions pushed out here. And if all of these guys are accumulating out in this area, what's happening to this area? It's becoming very salty. What's water going to want to do? He's going to want to go to the areas of where salt is. So where's water going to start flowing? It's going to start flowing out into this area here by a type of obligatory water reabsorption, right? Now, you might be wondering, how does this water get out? What are these blue channels? Because I didn't just draw them for no reason. They have to have a specific type of name. They do. These channels here are called aquaporin type 1. And the only reason I'm mentioning that because we'll talk about the collecting duct and we'll talk about aquaporin 2 and 3 and 4. But aquaporin 1 is these channels that are always open in the descending limb of the loop of Henle. And as this actual filtrate is going down, consisting of a lot of water, what happens is, as it's going down, the ascending limb, as it's going up, it's pumping out sodium, potassium, and chloride ions. And the concentration of the salt is increasing in this area. So as this is going down and this is going up, it's making the medulla really, really salty. So what happens to the water? It wants to move out of the descending limb and into this medullary interstitial space. So you start losing water passively. What is that called whenever as the ascending limb is going up this way and it's pumping out sodium, potassium, and chloride? As it's going up, it's doing that. And while water is going down, water's following the salt out into that medullary interstitial space. That mechanism is called, it's right over here, it's called the counter current multiplier mechanism. Okay, so one more time, let's explain the counter current multiplier mechanism. As this actual filtrate is moving through this area, I should mention something also. You're probably wondering, well, can't salt and stuff leave? No, because the actual descending limb of the loop of Henle is completely impermeable to solutes. What does that mean? That means salts can't go out and they don't really come in either. Okay? So again, one more time. As this is actually flowing down, the only thing that it's permeable to is water. It's impermeable to solutes. On the ascending limb, it's the exact opposite. It is completely impermeable to water, but only permeable to solutes. So what is it doing in this area here, in the ascending limb? It's pumping out sodium, potassium, and technically 2 chloride ions, right? 2 chloride ions. And as that's doing that, it's pumping this sodium, potassium, and 2 chloride ions in this area and making it really, really salty and concentrated and very, very hypertonic. As the actual descending limb of the loop of Henle is going down, what happens to the water? It wants to be where the salt is. So it leaves the descending limb and goes out into the medullary interstitium. And what happens then? If we're losing a lot of water, what would it be? Let's say it takes this turn. The filtrate takes the turn here, and it's going to get ready to go into the ascending limb. What do you think the osmolarity will be in this area here? It started off at 300, but then it gets saltier as you go down. So technically, if water was flowing out here, let's say it was 300, right? And it's almost about 300 there. The water is going to want to flow out into this area where the salt is until it's about 500 milliosmoles. Same thing here. It's about 700. So water is going to want to flow out of the actual kidney tubule, the descending limb, out into this area until the kidney tubule's plasma osmolarity is about 700 milliosmoles, right? Let's keep going down. Over here in this area, it's going to be about 900 milliosmoles. Water is going to want to keep flowing out into this area until his osmolarity in the kidney tubule is equal to about what? 900 milliosmoles. What do you think will continue to keep happening? Water will continue to keep going out into this medullary interstitial space until whenever it takes that turn and gets ready to go up, it is now 1,200 milliosmoles. What is 1,200 milliosmoles? Is that a pretty high number? I would say that's a lot. So if it's a lot, if it's a high osmolarity, what does that mean it's really, really rich in? It's rich in a lot of solutes like sodium and chloride and very, very low in water. So we're going to say then that this plasma osmolarity of the filtrate is now hypertonic to what? Hypertonic to the plasma osmolarity of 300 milliosmoles. Okay? Now, as we go up, it's really, really concentrated. But then what does it do when it's in the ascending limb? It's pumping out sodium, potassium, and chloride ions. And it continues to pump out sodium and potassium and chloride ions. When it goes away, what was it pretty much pumping out? It was only pumping out salt, right? Sodium, potassium, 2-chloride anions. No water was leaving. It actually goes until when it actually moves up to this area, it's almost equal to 300 milliosmoles. It's approximately around like 200 milliosmoles about when it gets into the distal convoluted tubule. So now again, one more time. As it's going down, it becomes, at the end of it, it becomes hypertonic. Why? Because you're losing a lot of water out into the medullary interstitial space. Why are you losing it? Because as the ascending limb is taking the filtrate up, it's pumping sodium, potassium, and 2-chloride ions out here through this cotransporter, making the medulla really salty. That's why you get the countercurrent multiplier mechanism. As it's going up, though, it's pumping the salt out, so it loses a lot of salt, or solutes. So the osmolarity, when it gets up to this point here, which is called the distal convoluted tubule, it's a little bit less than 300 milliosmoles, about like 200, okay? What is that called whenever your osmolarity is really low? Let's look. If the osmolarity is really low, that means that you don't have a lot of solutes and you have a little bit more water in that area than you do solutes. So you have a low osmolarity. That's referred to as hypotonic. So now, as the fluid leaves the ascending limb of Loop of Henle, it now is considered to be hypotonic with comparison to what? The plasma osmolarity of 300 milliosmoles. Okay? And again, it can range. I could say really the range is about from 120 to about 200 milliosmoles, really. Okay? And then the filtrate, we're going to the DCT, which we'll talk about next. But I got to talk about this guy now, the VasoRecta. So the VasoRecta is really important, okay? The VasoRecta is a branch of the paratubular, it's actually a paratubular capillary in the medulla. And so it's a branch of the efferent arteriole. Now, we refer to the VasoRecta as what's called the countercurrent exchanger. Countercurrent exchanger. Okay, so let's go back to the osmolarity. What was it as we were moving down? Let's assume that it was about 300 right here. Then as we moved down, it went to about 500. Then as we go down a little bit more, it went to 700. Then it went to 900, and then it goes to about 1,200, right? So that's our renal medulla. That's the medullary interstitial gradient, right? The plasma osmolarity gradient. So, you know, blood flow through the VasoRecta is really, really sluggish. It's really, really slow. It's moving really, really slow. And as this blood is flowing through this area, what's happening to the concentration out here? It's getting really, really salty. So what's really, really high in concentration out here? You're going to have a lot of sodium ions in here. You're going to have a lot of chloride ions in here, right? So as it goes down, it picks up a lot of sodium chloride. So it picks up a lot of sodium chloride and more sodium chloride as it's going down. But at the same time, if it's really, really salty in this area, and there's water coming from that area, so there's going to be water. Where's water going to want to flow? We've already talked about this. It's going to want to flow from areas of low plasma osmolarity, right, or low osmolarity to high osmolarity, where there's a lot of salt. So water is going to come out here to where it's salty. And if it's moving out here to where it's salty, look what it's doing. It's moving the water out, and it's bringing the sodium chloride in. But then look what happens. It makes this turn, and it comes back up. Now, as the plasma osmolarity starts decreasing, what are you going to want to do then? You're going to want to get rid of the sodium chloride. So now you're going to start pushing that sodium chloride out. And when it leaves, so it's about 300 going in, so it's about 300 milliosmoles going in. It's approximately about 325 milliosmoles leaving, almost exactly equal. So again, you're pushing out the sodium chloride. But look what else happens. As you're going up, the sodium chloride is actually going to get pushed out, right? So it's actually going to get pushed out. The water is going to move back in. Okay, so the water is going to move back in. What is the significance of this? The whole purpose of having the sodium going in, the salt going in, and the water going out, going down the VasoRecta, and then the exact opposite occurring, you're going up the VasoRecta where it's pushing the salt out and bringing the water in, is because, imagine this. Let's imagine all this area right here. It's really, really salty, a lot of sodium, a lot of chloride. If we have a lot of fluid coming through here, and let's say it washes away that sodium chloride, you're not going to be able to maintain that nice medullary interstitial gradient. So all this VasoRecta, or the countercurrent exchanger, is doing is it's preventing the rapid removal of sodium chloride. So it's not actually developing this medullary interstitial gradient or this countercurrent multiplier mechanism, right? It's not the one generating it, it's maintaining it. So again, one more time, what is the function of the countercurrent exchanger? As it's going down, it's picking up salt and getting rid of water. As it turns the corner and goes up, it gets rid of the salt and brings in water. And it takes a little bit more salt away. What's the significance of this? To prevent the rapid removal of sodium chloride from the medullary interstitium so that you can maintain a nice gradient, a nice salinity gradient. Okay? That's one function. You know what else? It carries oxygen. These cells, they depend upon oxygen. So he's also going to be delivering oxygen and certain types of nutrients to these tissue cells also. So that's another function. So two functions of the VasoRecta. One is helps to maintain the medullary interstitial gradient by preventing the rapid removal of sodium chloride from this medullary interstitium. And the second one is it provides oxygen to the tissue cells. And again, remember that the VasoRecta has a very, very sluggish blood flow. All right, so let's go ahead and sum everything up then. So let's get this out of the way. So let's go ahead and start with what's happening in the descending and what's happening in the ascending. So if you remember, descending limb, what was the whole purpose here? Remember that it was water, permeable, meaning that water can move in or it can move out, right? And it was solute, impermeable. So no salts can leave, right? So no salts can leave here. So it's actually going to be water permeable, solute impermeable. Now, what were those proteins that were present within the actual membrane? Those were called aquaporin 1. So aquaporin 1, right? And what was aquaporin 1 allowing for? It was allowing for the water to move out where? Out into the medullary interstitium, right? So interstitium. Why was it moving out into the medullary interstitium? Because remember what was happening in the ascending limb? In the ascending limb, you had what? You had that sodium, potassium, 2-chloride cotransporter. And what was that sodium, potassium, 2-chloride cotransporter doing? It was pumping sodium out. It was pumping potassium out, and it was pumping 2 chloride ions out. Out where? Out into the medullary interstitium. If it pumps it out into the medullary interstitium, what's it going to do to the medullary interstitium? It's going to make it really salty. If it's really, really salty on that medullary interstitium as it's going up, what's it going to do? It's going to want to pull the water out into that area. What is that called whenever the water moves out into the area of the medullary interstitium as it's going down, while as the ascending limb is moving up, it's actively pumping out that sodium, potassium, and the chloride. That whole event is called the countercurrent multiplier mechanism. And who was maintaining this salinity gradient, this salty gradient within the actual medullary interstitium, preventing the rapid removal of the salt and providing oxygen to the tissue cells? It was the VasoRecta, our countercurrent exchanger. I actually told you I was going to tell you something about that potassium, didn't I? Remember I told you that some of that potassium stays out here? What happens is some of that potassium gets released out. But what happens is some of that potassium actually gets pushed into the lumen of the filtrate. When it gets pushed in, it creates a depolarization of the inner side of the membrane, this ascending limb. What's present in this area is calcium and magnesium. Because there's a lot of positively charged or depolarization of this inner lining of the actual luminal membrane, they can't move out. They can't actually get out of this area. Usually they want to be able to get out of this area, right? What happens is, because they're actually trying to get out into this area, they get bounced around because of these positive charges, right? Because they're repelled. What eventually happens is they get bounced around, boom, 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 and then move right through this area right here. They actually move right in between these cells. What is that called whenever the calcium and the magnesium moves in between the cells? That's called paracellular transport. So the calcium and the magnesium could also undergo reabsorption into this actual medullary interstitial space by paracellular route. All right, so we covered a lot of information, guys, a lot of stuff with respect to the loop of Henle. We covered the countercurrent multiplier mechanism, the countercurrent exchanger, a lot of different information. I really hope all of it made sense. I hope you guys enjoyed it. If you guys liked it, hit that like button, subscribe, leave any comments. We'd appreciate it. We really want to be able to help you guys out and make a difference. All right, Engine Nerds, until next time.